this video we will discuss about glycans and its relationship with the autophagy. So we will cover two different parts. First one is that how these glycans can trigger the intrinsic death, at the same time how these glycans can help in this autophagy of the viruses. So we will start with these glycans and the intrinsic cell death relationship. So this relationship has been explained through this mitochondria, uh, to the interactions of glycans with this mitochondria. So we can see that the BCL2 is been shown in the cytosols, and this BCL2 can interact with this TBD and LND treated cells. Uh, in TBD and LND treated cells, uh, this galectin 7 can interact with this BCL2 to form a complex. And even though the cells are not treated with TBD and LND, the galectin 3 can interact with this BCL2 to form a complex. And this complex formation will lead to the release of the cytochrome C within the cytosols. And this cytochrome C, when leaks into the cytosol, it will trigger the intrinsic cell death. So this is how these glycans can play a role towards this intrinsic cell death within the intrinsic cell death, with the help of this modifying the membrane of this mitochondria. At the same time, it will also cause this signaling pathway, that is SMAC and diablo signaling pathways. So this is about intrinsic cell death and the glycans. Now let us understand how these glycans helps in the autophagy of the viruses. So this thing has been explained with the help of this semolina coated vollicle containing vacuole, not molecule, vacuole. So somolina containing vacuole. This is the kind of virus. So what happens in this case? The galectin A plays a role. So when this SCV reaches into the endoplasmic reticulum, then this glycans, the galectins A will interact. Galectins A, when the SCV reaches into the endoplasmic reticulums, and they are decorated with the glycans. At the same time, the galectins A helps them decorate with the host pertaining glycans, and thereafter it will form a complex with this NDP2 and LC3. And once they form a complex, SCV forms a complex with NDP2 and LC3, then they can be directed towards autophagosome, where they will go to this autophagosome process. And empty vacuoles can go into the lysosomes, where they can be recycled. So and this is one of the way by which this autophagy can trigger, and it will this galectin A can act in a cytoprotective manner. At the same time, what can be seen is that uh, if there is a deficiency of this GMAP 210 within the endoplasmic reticulum, then it will lead to the decrease in the cargo of glycoproteins that helps in the transport of the proteins from endoplasmic reticulum to the Golgi. And this decrease in the transportation will not only leads to the increase in the amount of proteins within the endoplasmic reticulum, that will trigger ER stress, and thereby it will trigger other signaling pathways such as IRE1, PT1. And if this ER stress is beyond the limit, then this ER stress might also trigger apoptosis or autophagy. So this is one of the condition that occurs when there is a deficiency of GMAP 210, and that will lead to the decrease in the amount of alpha mannose, and that will cause the ER stress. And so this is about the another way by which these glycans can trigger autophagy, or deficiency of the glycans can trigger autophagy. Let's come to the main topic. Okay, and what happens after this case is that in these conditions where there is a deficiency of GMAP 10, then we have discussed how it will trigger ER stress. And there is another condition where there is a deficiency, or this mannose-6 phosphate is not working properly. If the mannose-6 phosphate is not working properly, then it will lead to the improper glycosylation from the Golgi apparatus, that is from the trans-Golgi network, and thereby it can interact. This improper glycosylation of the proteins can interact with this autophagosome, and that will lead to the uh, polyubiquitation of the proteins, and that will it helps them to divert towards this autophagosome, or that in the lysosomes, where the degradations of the proteins can occur, and the amino acids can be recycled for further can be recycled for new formation of the proteins. So I hope you got an idea about how these glycans and autophagy are related, and how the glycans can help either in the interstitial cell death, or it can also helps in it can also helps or works as a cytoprotective protein in cytoprotective intermediate proteins, for example this galectin A, where it can divert this SCV towards this autophagosome. If you need to understand more about this glycans and autophagy in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.